concept in kingdom planting is the different types of life cycles and alternation of generation. So here in plants, life cycles and alternation of generation. So in plants, each and every cell, it may be a haploid cell or it may be a diploid cell that undergoes mitosis. So it may be a haploid or it may be diploid that undergoes a cell division called as mitosis. Now, the ability of these cells, either which may having a single or a two sets of chromosomes, that capable of undergoing this equational or homeotypic cell division called as mitosis, that result in the formation of a haploid or it may be a diploid plant body. So that is one of the important attribute of these plant cells. Now, when that undergo mitosis, that too in case of a haploid cell, when that undergo mitosis to produce haploid cell only, that means that develop into a gametophyte. If the diploid cell which undergoes mitosis, again that retains the same number of chromosomes and that develop into a sporophyte. So that is gametophyte as well as sporophytic plant bodies. So generally in all the sexually reproducing plants, those plants which shows a advanced mode of reproduction, that is sexual mode of reproduction, where there is a production of gametes. In that condition, always we can observe there is a alternation of generation. One phase in the life cycle of an organism alternates with the another phase. That is commonly called as alternation of generation. Now, so here in case of plants we can observe different types of life cycle, that is haplontic life cycle and diplontic life cycle and haplodiplontic life cycle. These are the three types of life cycles we can observe in case of plants. So what is the haplontic life cycle? This haplontic life cycle is the characteristic feature of thallophytes, the two in case of algae. that too in majority of the algae, haplontic life cycle is present. Now, in case of haplontic life cycle, the sporophytic generation is single celled. The sporophytic generation is what here? The sporophyte is single cell. Single cell, that is represented as what? That is zygote. And this zygote, now that undergo mitosis. Zygote is a single celled one that undergo mitosis to form the sporophyte, and this sporophytic generation is very short-lived. Very short-lived, that is single celled form, and it always that depends on the gametophyte. That always depends on what? Gametophyte, and this is always short-lived. And this sporophyte again that undergoes meiosis to form a gametophyte. That means meiosis means that forms what? Haploid cells. That the haploid cells develop into gametophyte, and this gametophyte is multicellular, photosynthetic, and it is independent. And this is the dominant phase in the life cycle of this algae. Only the sporophytic generation is represented as a zygote, that is single cell. Immediately that undergo what? Again meiosis to form the haploid gametes, that is the haploid cells, that gives rise to a gametophyte. That is the predominant plant body that is called as haplontic life cycle. What is haplontic life cycle? Haplontic life cycle is one where the haploid phase of the plant is very dominant, that is gametophytic phase is predominant, and that is long-lived, which is multicellular and photosynthetic in nature, and it is always independent and free living. Then the sporophyte is represented by a single celled zygote. Again that single celled zygote which develop into a sporophyte, but which is always dependent, non-photosynthetic, and that undergo meiosis to form again the haploid cell which develop into a gametophyte. That sporophyte produces what? Haploid spores that develop into a gametophyte. Therefore, this life cycle is called as haplontic life cycle. And second is called as diplontic. So, as the name itself indicates, diplontic life cycle is the characteristic feature of all vascular plants that include mainly the pteridophytes, gymnosperm, especially the higher vascular plants we see. Now, in case of diplontic life cycle, the dominant phase is diploid sporophyte, which is the dominant phase, that is the diploid sporophyte, and it is multicellular. It is multicellular, well differentiated, photosynthetic and independent. And this sporophyte, this produces spores by means of meiosis, and that now mainly develops into a male as well as female gametophyte. And again, the major components of the female and male gametophyte fuse to form a diploid zygote, and that is a diploid zygote undergo mitosis, and that gives rise to a multicellular predominant sporophyte. So here this dominant phase is diploid sporophyte, which is multicellular, photosynthetic and independent. It produces spores by means of meiosis. That means which becomes what? Haploid. That haploid that gives rise to a highly radiosed. This haploid gives rise to reduced male gametophyte, that is commonly called as microspore, or we can say coral grains, and reduced female gametophyte, reduced female gametophyte, that having male and female gamete. Now the fusion of that male and female gamete, again that gives rise to a zygote. That zygote now undergoes mitosis, which results in again sporophyte. So the major plant body is diploid, which is called as sporophyte. Therefore, this kind of life cycle is called as diplontic life cycle. Here, gametophytic generation is highly reduced and it is very short-lived and it is non-photosynthetic. Therefore, this kind of life cycles are called as diplontic life cycle, generally in higher vascular plants. Then the third type of life cycle is the intermediate type of life cycle, that is called as haplodiplotic life cycle. Haplodiplotic life cycle. Now, this is the intermediate type. That too in case of bryophytes. So bryophytes are called as amphibians of the plant kingdom, where the predominant plant body is haploid. That too haploid, that is called as what? Gametophyte. And this gametophytes that produces gametes during the gametogenesis, and that gametes produced by the male as well as female sex organs, which undergo fusion. Gametes that undergo fusion, that is syngamy, resulting what? Zygote. But this zygote, which is diploid, that develop into a sporophyte, but that sporophyte is depending on the gametophyte, because the sporophyte is always fused cell, highly reduced, and that dependent on the gametophyte because that is present inside the female gametophyte. That present inside this gametophyte. Therefore, we can say this is depending on the gametophytes. And that sporophyte again undergo meiosis and which produces what? A haploid spores. Later on that develop into a gametophyte. So this kind of what life cycle here we can say that is haplontic. And in case of pteridophytes, so here mainly haploid phase alternates with the diploid. And in case of pteridophytes, where the major plant body is a sporophyte, that sporophyte undergo reductional cell division, that is meiosis, and produces mainly the spores which are haploid, and that spores on fusion that forms a zygote. That spores that form a zygote. Now the zygote undergo again what? Meiosis. So in that way that continues. But here mainly the sporophyte which produces the spores, that is the haploid spores, and these spores generally microspores or it may be a megaspores, and that microspores and megaspores that develop into gametophytes. This develop into what now? Gametophytes, and these gametophytes are called as prothallas. And this is always photosynthetic and it is independent. This is also independent, this is also independent. Here we can observe both the diplontic as well as haplontic. Therefore, this kind of life cycle is called as haplodiplontic life cycle. So this is intermediate type of life cycle. So this completes the metaphyta, or it is called as kingdom Plantae. So the next to this kingdom Plantae is the kingdom which includes multicellular organisms which are heterotrophs without any cell wall, that are called as what? Animals or metazoa, that is animal kingdom. Now, so here more than or around a million of animals are identified. Strictly speaking, 10 lakh number of species till now are identified by the zoologists on this earth. So therefore the need is created to classify all these discovered animals. Now this classification is very important. It is mainly to assign the animals in a systematic position. The scientist can assign the animals into different groups. They can assign that particular species in a particular rank based on certain features. That is one of the importance of this classification as well as assigning of the animals. Now, these animals, that is the different species of animals that greatly differ in its form as well as structure. In spite of that differences, even though that animals differ in their form as well as structure, but all the animals basically or fundamentally that show certain common features. Even though that differ in different forms, but that show some fundamental similarities or common features. Now, what is the criteria for the classification of these animals? So, the next concept is what? The criteria for the classification of animals. On what basis zoologists they classify the animals or assign the animals? So basically the first criteria is organization, that is cellular organization. Cellular organization. And second is body plate. Third is germ layers. And the fourth one is based on symmetry of the animals. Then based on coelomic cavity, based on circulatory system, based on segmentation, then based on the presence of notochord. All these are the basics or criteria for the classification of animals. So under that, the first one is cellular organization. Now, even though all the animals which comes under the kingdom Animalia that are called as metazoa, later formed animals which are commonly multicellular. Even though the animals are multicellular, but that shows different type of cellular organizations. So under that, some animals that shows only cellular level of organization without tissue grade. Even though the animals are multicellular, but the body that composed of the group of cells, but that group of cells which are not organized to form the tissues. Here the cells are loosely arranged. The cells are which kind of metazoa, multicellular animals, which shows the loosely arranged group of cells without tissue organization that are nothing but parazoa, that is represented by one phylum called as porifera, or we can say sponges. Parazoans are multicellular, but the cells are very loosely arranged, that are not organized in a particular pattern to form the tissues. Only that shows cellular grade of organization, and that cells are organized for the different functions. That loosely arranged cell aggregations meant for different functions. That too in case of parazoa, that are nothing but porifera or sponges. Then after that cellular grade in the course of evolution, that group of cells which are cells which are very closely arranged to form a aggregations, that group of cells organized to form a tissues. That group of cells having a common origin, that shows a common structure and also the similar type of function, that constitute what? Tissue grade of organization. So here the group of cells aggregate to form the tissues. So that animals are usually exhibiting what? Tissue grade of organization. So which are those metazoans which comes under tissue grade of organization? That are nothing but coelenterata as well as coelenterates as well as ctenophorans, that shows tissue grade of organization of the body. But in case of porifera or in case of the parazoa, only there is a cellular level, loosely arranged cellular level of organization. So next to this, now in the course of evolution, the group of tissues which organize to form a specific structure called as organ. So first is cellular level, next is tissue grade, next is what? The tissues which organize to form the specific structure, that is organ grade of organization. So which are those metazoa that exhibit only organ grade of organization? So that are nothing but platyhelminthes. That are nothing but what? Platyhelminthes. Now again in the course of evolution, that different organs that organize to form a specific system meant for specific function, that level is called as organ system level of organization. Organ system grade of organization, that is starting from Helminthes up to the chordates. So in this way, based on the cellular organization, we can classify the animals, whether which show only the loosely arranged cellular level or tissue grade or organ grade, or it may be organ system grade of organization. So the next criteria, the next one is, that is body plane. So first is what? Cell organizations. That is cellular organization. This is one. Second is body plane. So here based on body plane, how we can classify the animals? Whether the animals which shows blind sac body plane or that may show the tube within the tube body plane, in that way we can classify. So the body based on body plane may be blind sac body plane, or it may be a tube within tube body plane. Tube within tube body plane. In case of this blind sac body plane, and even there is cellular body plane. Cellular means where the cells. So that also we can keep cellular body plane. Here, as already stated in case of parazoa, even though the animals are multicellular, but that cells are loosely arranged, and only we can observe the cellular level of organization of the body. So here these cells which are organized for the different functions of the body, these cells that are loosely aggregated for the different functions of the body, that is called as cellular body plane. What is cellular body plane? Where the loosely arranged group of cells which are organized for the specific function to form the body of an animal is called as cellular body plane. Blind sac body plane is one where the animal body cavity is provided with the animal that having a cavity, that body cavity having only single opening. So that having single opening for that body cavity, and that single opening that meant for both ingestion as well as egestion of the components, that is called as blind sac body plane. Blind sac body plane is one where the body cavity that having a single opening meant for both intake as well as what? The egestion. That is called as blind sac body plane, as in case of coelenterate and Pinofora. This is in case of sponges. Tube within tube body plane. So the tube, this tube is developed by the body wall. Outer tube is formed by what? Body wall. And this encloses another tube. This is mainly a digestive tract. So here the body within, that is our tube within tube body plane is one where there is a outer tube which is formed by the body wall that encloses another tube that is nothing but digestive tract. So this is another type of body plate. That this is starting from the plotted helmet base up to cortex. All that shows what type of body plate, tube within tube body plate. So based on body plate, so that is we can usually classify the animals which are based on body plate. The animals can exhibit cellular body plate, blind sac body plate, tube within tube body plate. And the third what criteria. Now, usually, so here some animals that shows open type of circulatory system and some shows closed type of circulatory system. Open type of circulatory system is one where the blood is pumped by the heart. So whatever the blood pumped from the heart that comes out from the heart and enters into the body, where each and every cell and tissue of the body is always it is dipped in the blood. That means, so in case of open type of circulatory system, the blood pumped from the heart into the body spaces, into the where body spaces where each and every cell and tissue is bathed in the blood. There is no definite channel to carry the blood. So the blood flows through the cavities or spaces where the cells and tissues are bathed. That is called as open type of circulatory system, as in case of arthropods. Closed type of circulatory system. What is closed type of circulatory system? So as the name itself indicates, closed means the blood pumped from the heart is carried by definite channels. Definite channels which are nothing but blood vessels in the form of arteries as well as veins. The blood is carried by blood vessels, not through the body spaces. Here, the cells or tissues are not bathed in the blood. That is the difference between open and closed type of circulatory system and this closed circulatory system as in case of vertebrates. Now, so the next is based on germ layers. Based on what layers? Germ layers. What are these germ layers? During the development of the embryo, during the embryonic development, so the germ layers formation takes place, which are in the form of ectoderm, endoderm and mesoderm. These three layers are called as germ layers that are developed during the embryonic stages. Now, so based on these germ layers, the animals are classified into, based on germ layers, animals are classified into diploblastic and triploblastic animals. Based on germ layers, the animals are classified into diploblastic animals and triploblastic animals. Here as the name itself indicates diplo. Diplo means what? 2. Triplo means 3. Diploblastic animals are those which having only 2 germ layers in the embryonic development. How many germ layers you can observe? Only 2 germ layers, that is outer ectoderm as well as outer ectoderm and inner endoderm. Outer ectoderm and inner endoderm. In between that there is a mesoglia. In between that there is a jelly like what? Mesoglia. This kind of animals are called as what? Diploblastic animals. As in case of ctenophorans as well as coelenterates, that having only 2 wall layers, that is germ layers, ectoderm and endoderm. And in case of triploblastic animals, so triploblastic animals are those which having 3 germ layers, outer ectoderm and inner endoderm, and in between that there is a distinctive layer called as mesoderm. This is ecto, this is endo, and this is mesoderm. In between the ectoderm and endoderm there is a definite germ layer called as mesoderm. Such animals are called as triploblastic animals. Starting from platyhelminthes up to the chordates, all animals are triploblastic. This is very important. Differentiate between diploblastic and triploblastic. Diploblastic animals are those which having 2 germ layers, outer ectoderm and inner endoderm, and there is a undifferentiated mesoglia in between ectoderm and endoderm. But in case of triploblastic animals, there are 3 distinctive germ layers, outer ecto, inner endoderm. In between that there is a middle distinctive germ layer called as mesoderm. This kind of animals are called as triploblastic animals. So the first is based on what? The cell arrangement, cellular organization. Second is what? Based on body plane. Then is based on what layers? Germ layers. Then is based on symmetry. Symmetry indicates what? The plane of body division to give the different hands. So here based on symmetry, animals may be asymmetrical, radially symmetrical and bilateral symmetry. So based on the what symmetry, 3 kinds of what animals we can identify: asymmetrical, radially symmetrical and bilateral symmetrical animals. Asymmetrical animals are those when the body plane of the division passes through the center. If the plane of division passes through the center, if the knot gives equal halves, it is called as asymmetrical. It is irregular shape. That knot gives equal halves. If we cut the animal at any plane, if the what plane of division passes through any what axis of the body, if that knot gives the 2 equal parts, that is called as asymmetrical as in case of sponges. Radially symmetrical animals are those when the plane of what division passes through the at any plane. The what division passes through any plane of the body axis that gives two equal halves, that is called as radially symmetrical. Radially symmetrical animals are those which gives two equal parts when the plane of division passes through the any axis of the body. Bilateral symmetry is one which gives two what equal halves when the plane of division passes through the central axis. So when that gives two equal halves, when we cut the animal, that plane of axis passes only through the central axis, that gives two equal halves. It is called as bilateral. When the body plane of the division passes through any plane of the body axis, that gives two equal halves, that is radially symmetrical. This not gives any, that not produces equal halves when the plane of division passes through the any plane of the body axis, that is asymmetrical. Now, radially symmetrical animals are coelenterate and ctenophora. Bilateral symmetrical animals starting from Platyhelminthes up to the carpets. All that shows bilateral symmetry. So asymmetrical animals are those when we cut the animal to any body plane that not gives two equal halves. When we cut the animal, the plane of the division passes through any axis of the body that gives two equal halves, that is radially. If that gives two equal halves, right and left half, only when it passes through the central axis of the body is bilateral symmetry. Then, based on the coelomic cavity, what is coelomic cavity? What is coelom? Coelomic cavity means it is the cavity present between the body wall as well as alimentary canal. Body cavity present between the body wall as well as digestive tract, or we can say alimentary canal, is called as coelomic cavity. Cavity present between the body wall and alimentary canal, that is digestive tract, is called as coelomic cavity. Now, based on this, there are three groups of animals: A. Coelomates, pseudo coelomates, and eucoelomates. So, based on this coelomic cavity, we can classify animals into three groups. One is acoelomatic animals are acoelomates, pseudocoelomates as well as eucoelomates. What are these acoelomates? Acoelomates are those animals where there is no body cavity. There is no body cavity between the body wall as well as alimentary canal. There is no any cavity present between the body wall and the alimentary canal, are called as acoelomates. as in case of platyhelminthes, flatworms. Pseudocoelomates means those animals which having a cavity, body cavity in between the body wall and alimentary canal, but that is not lined with mesoderm, which is not lined with mesoderm, but that mesoderm is scattered in the form of pouches. Which is scattered in the form of pouches? Mesoderm. That kind of animals are called as pseudocoelomates. Pseudocoelomates are those where there is a cavity between body wall and alimentary canal, but that body cavity is not lined by the mesoderm, and mesoderm is scattered in the form of pouches. Therefore, that are called as pseudocoelomates, as in case of Helminthes, that is roundworms. Helminthes. Next, what are eucoelomates? EU means definite, true, distinct. Eucoelomates are those animals which having a coelomic cavity, that is body cavity, between the body wall and alimentary canal, and that is lined by mesoderm. Whenever the body cavity present between the alimentary canal and body wall, which is lined with mesoderm, then that is called as true coelomic cavity. Those animals which having a coelomic cavity are called as eucoelomates. Pseudocoelomates means those animals where there is a body cavity but which is not lined by mesoderm, but mesoderm is scattered in the form of pouches. Acoelomates means those animals without any body cavity in between the body wall as well as alimentary canal. Now, eucoelomates starting from the arthropoda up to the chordates all comes under eucoelomates. That is based on coelomic cavity we can classify the animals into acoelomatic animals, pseudocoelomatic animals as well as eucoelomatic animals. Then based on what? So after coelomic cavity is based on segmentation. So it's based on segmentation or metamerism. So based on segmentation, the body of the animal, well, that divided into number of repeated segments. When the body is divided into number of segments, this is called as segmentation or metamerism. That is division of the body divided into number of units or segments. That is external segmentation corresponds with the internal segmentation. So externally as well as internally the body is divided into number of repeated segments. That is called as segmentation or metamerism. Starting from the way here usually arthropoda, annelida, all that shows metamerism or segmentation. That is based on segmentation we can classify segmented animals and unsegmented animals. Then is based on notochord. Notochord is highly flexible rod which is derived from mesoderm. Flexible rod derived from where derived from the mesoderm. Those animals which having notochord are called as chordates. Without notochord are called as non-chordates. So this is also one of the criteria for the classification of what animals. Now, so the first is what? Cellular organization, body plate, germ layer, symmetry, cenomic cavity, segmentation, as well as based on notochord. By all these, now we can classify the animals and we can assign them into a particular position.